Good morning, everyone. My name is André Marette. I have the pleasure and the privilege to chair this session this morning. We have three amazing speakers. We're going to have a roundtable discussion after that. So this symposium is organized by the Danone Institute, and the title of the symposium is Nutritional Targeting of the Microbiome for Healthier Lifespan. So a few words about the Danone Institute. It was established in 1991. It is a nonprofit organization that is committed to advancing science and fostering collaboration in the field of nutrition and health. In 2025, the Danone Institute opened a new chapter, which is dedicated to closing food and nutrition gaps in adults to foster healthy aging across the lifespan, with the ambition to reframe the narrative of aging. As you can see on this slide, the Institute is composed of several independent organizations in the world, in 3 continents. Those local roots of the Danone Institute are very important. They operate into a global framework, into implicating or fostering the research about nutrition and healthy living. That's the governance of the Institute. It's composed of several investigators. I'm one of the external experts together with Paul Scheel from Glasgow, and also Lisette de Groot from Wageningen University in the Netherlands. The president of the Danone Institute is Dr. Simeoni. And we have this global scientific board. The overall scientific direction is aligned, of course, with the scientific roadmaps, with the SHAME framework, and fostering a multidisciplinary international community of leading experts and partners. Now, there's 2 main core activities that we are trying to achieve with the Danone Institute. We are funding research through grants. We have an award, very prestigious one, the first one of the Danone Institute International Prize, in partnership with Nature Group and the French Foundation for Medical Research. This year in November in Paris, we'll be releasing the first award, so the nominations are currently being evaluated. And of course, in the research funding, we also enable partnerships with different leading institutions in the world, like, for example, the Chinese Nutrition Society or Harvard Medical School. We also consolidate science in different parts of the world with different institutions. We are building scientific consensus through reviews and roundtable, like the one we're going to have at the end of the symposium. We publish and amplify evidence-based content. And finally, what we're doing today is to convene scientific events and symposia in different places of the world, like in Mexico, here in Seoul, and later in Yokohama in Japan, where we're going to have another symposium at the ICPH-ICOF meeting in October. So yesterday we had a great talk from Rob Knight, and Rob has published this review a few years ago. I really like this figure because it shows the diversity and the change, the succession of different microbiome during life, from the cradle to the grave. And when you see these shifts in the microbiome with age, I see opportunities. There are tipping points during life where you can intervene, hopefully with nutrition or biotics, in order to try to change the aging trajectories, not only to improve longevity, but especially healthy longevity. This is all what we want. We want to live longer, but especially healthier. So today I have the privilege to chair the symposium with 3 distinguished lecturers, very well-known speakers in the field of the microbiome. I will mention a little bit more about them during the presentation, but we have Professor Dr. Iran Elina from the Weizmann Institute of Science in Israel. Then we have a talk from Jens Walter from the College Cork, Ireland. And then we have Professor Sean Gibbons from the Institute for System Biology, USA, which will give a 20 minutes presentation each, followed by 5 minutes of questions by the audience. And then we'll move to the discussion, the roundtable discussion, and we'll be joined by Professor Fu and Professor Lei, who actually both of them gave wonderful talks yesterday if you were there. So we're very pleased to have them join the discussion later on. So without further ado, in the interest of time, I will invite our first speaker, so Dr. Eran Elinav.
Thank you very much for this kind introduction, and many thanks for inviting me. It's always a pleasure to come back to Seoul. As you've heard, my two groups in Israel and in Germany have been studying host microbiome interactions for the past 2 decades. And referring to today's aging and lifespan topic, I think it's very fair to say that in the expanding longevity field, it is clearer and clearer that microbiome features play incredibly important potential roles in major hallmarks of healthy and unhealthy aging. And even more important aspects of this is that we've been discovering this expanding field that practically all the diseases that become more and more prevalent during aging are also tightly associated or correlated to the microbiome. And this, as you've heard in this meeting and in others, is a very expanding topic of investigations. We in our lab pride ourselves on trying to go beyond correlations and associations in trying to understand causality and molecular mechanism. We use a variety of descriptive and experimental modalities in an increasing toolkit that enables us to go from correlation into causation. And we believe that with such know-how, the field would gradually be able to introduce interventions that will harness the microbiome towards prevention and treatment of disease. And in doing so, one of the hallmarks that have emerged is that one can treat the microbiome as a signaling hub. And I'm specifically referring to the gut microbiome, but this may also be true for other ecosystems. So it's a signaling hub that integrates into it many signals that come from within our human body. Our immune system, our genome impacts the composition and function of our microbiome, but also a very large number of environmental factors that surround us impact the microbiome. The microbiome perceives all of these signals, integrates them, and communicates them to the host. And we see our mission in life as trying to decode the alphabet of the language by which we and our microbes talk in health and what goes wrong when it's disturbed and predisposes to disease. And one of the major, maybe the major factor that regulates the healthy and unhealthy microbiome configuration in the gut is our diet. And given the focus of this session, I'll exemplify this by a couple of short vignettes from our own work in trying to decipher how features of diet impact the microbiome and how such impacts may signal to the host in health and in disease. So the first vignette relates to the composition of diet, the micro and micronutrient makeup of our different diets and how it impacts the microbiome. And when you look into everything that you and I do in our normal healthy lives when we try to come up with what we hope would be a healthy diet, it becomes clear that everything that we do for the past many decades is based in one way or another on a group of matrix that give different numbers to foods. For example, calories is such matrix, glycemic index is another matrix by which we give numbers to foods based on their ability to induce spikes in blood sugar level. And based on these numbers, we try to come up with what we hope would be a healthy diet. Now, a decade ago, in work that we performed together with Eran Siegel, who will also be speaking in this meeting, we discovered that this entire concept is inherently flawed, and the first eureka moment came when we studied in a very naive study how artificial sweeteners impact the microbiome and the human host. And part of that study involved giving a group of volunteers an identical amount of saccharin, or one of any other nutritive sweeteners, and following them for their spike in blood sugar levels. And we were surprised to find that half the people spiked the blood sugar levels when they were eating these seemingly inert sweet substances, and half of the people didn't care, which by definition told us that giving a number to saccharin could not make any sense, because if your response is opposite than mine, then the same value cannot be given to both of us. Or in other words, we need to start giving numbers to the individuals eating saccharin rather than to saccharin itself. We've then expanded this work and tested thousands and tens of thousands of real-life foods, and in all of them, the variability in people's response when they were eating the same exact food was unbelievably big, meaning that we need to shift from this matrix of measuring the food to a matrix that measures the human individual. And this was achieved in a very ambitious project. At the time, we termed it the Personalized Nutrition Project, in which we took a group of 1,000 people. We collected everything at the time that was possible to collect from them, including a very detailed microbiome analysis. We've measured the blood sugar level with a continuous glucose monitor. We generated personalized algorithms using deep learning and machine learning to try and predict a person's response to any given food. And we're happy to find that we could do this to a great extent using people's personalized data. We then engage in short-term interventions. You can see here an example of a continuous glucose monitor readout from a single person who was put through our process and then prescribed counterintuitive, personalized good diets and bad diets. The good diet could include, some chocolate, and the bad diet could include sushi. Until you measure, you don't know what to expect. And then they were asked to consume their bad or good diets. And you can see that during the week of consumption of bad diet, in red, you can see these spikes in blood sugar level. You can see that the same person, when he was put on the counterintuitive good diet that could include ingredients that you may consider to be unhealthy in green, these spikes flatten out. And when we revisited these deep learning algorithms in retrospect, they never asked for my permission on which feature to use. We were happy to find that some, not all of the features, include the gut microbiome. In fact, today, when over 100,000 of these people have been profiled, you can just take the microbiome and a more limited set of personalized features and get the same accuracy. This then led to a long-term randomized controlled trial in which pre-diabetic individuals, 200 of them, were randomized into the best of the best Mediterranean diet or this counterintuitive but data-driven personalized approach. They were followed for a whole year. And you can see on the bottom an example from 3 different glycemic readouts generated in this study. In red, you can see this Mediterranean diet effect. It's there, as was published for decades, but it's rather mild. But you can also appreciate that the personalized approach outperforms the one-size-fits-all approach by quite a nice margin. Now, this personalized nutrition approach also gradually carries global impacts. For example, if we go back to the sweeteners and microbiome impact on their variable effects on the human body, just a couple of years ago, following our studies, but also many others that followed, the WHO, the World Health Association, have issued its first ever global warning against the indiscriminate use of sweeteners, especially by populations at risk, diabetics, pre-diabetics, and so on and so forth. And today, this has given rise to a quite exciting field with many studies from other groups and other populations corroborating our principal findings. We are following up in trying to understand more on this in special populations such as children with obesity, and trying to very carefully apply the same method on non-metabolic disease such as inflammatory disease. And the NIH has initiated a flagship a couple of years ago in which lots of money is being invested in trying to expand this field. So this was an example of how we see dietary composition and its interactions with the microbiome as meaningful in human physiology and disease. An equally interesting feature of dietary microbiome interactions that we've accidentally discovered relates to the timing of our diet. And this was a part of a study which very naively tried to answer a question related to circadian activity. We know that every cell and organ in our body, changes very reproducibly its function along a 24-hour cycle. There's a Nobel Prize that was awarded to the person who discovered the clock in each of our cells. And since we regard the microbiome as a neglected organ, we asked ourselves whether the gut microbiome also features a very reproducible diurnal or circadian activity, which was very painful because you can imagine postdocs collecting poop every 4 hours from animals or from humans, and then doing this for consecutive days and sequencing them and so on and so forth. But the result was very exciting because we found that both the composition and the function of the gut microbiome features a very robust circadian activity. You can see on the left an example of one of the commensals. It goes up during the night, down during the day, up during the next night, down during the day. You can measure forever, and it would always be the same. On the right, you can see a few dozen of microbial functions that go up during the night, down during the day. Now this was very exciting, but it was also very strange to us because if you think about it, the gut microbes in the gut, they live completely in the dark. So how do they know that it's day and night? And so this really naive question resulted in another couple of years of work. And at the end, the answer emerged to be the timing of our diet. The microbes feel that we're eating and awake and change their behavior accordingly. And when we're asleep, we don't eat, and they feel that as well and change their behavior and composition accordingly. Just to give you an example of how in many experiments we figured this out, this is a mouse experiment. On the left upper panel, you can see a group of mice that are force-fed only during the nights and are fasted during the days. On the right side, you would see an identical group of mice that are force-fed only during the day and are fasted during the night. And when you sequentially sequence and characterize their gut microbiome a long time, you can completely shift their microbiome composition and behavior by a 12-hour shift, meaning that the exposure to different patterns of timing of diet determines this behavior. Now, you may ask, why is this important? It's important because from the beginning of the Industrial Revolution, after many millions of years in which we were accustomed as a species to abide to circadian activity, we woke up with the sun, we did whatever we did, we worked, and then we went to sleep when the sun set. After the Industrial Revolution, all of a sudden, for the first time, we beat the circadian activity. There are lights and electricity, so people started working in shifts. There are planes, we can travel across time zones and get severe jet lag, such as the one I'm suffering from. And with this chronic sleep-wake disturbance, came a very dramatic susceptibility of such shift workers, for example, to develop obesity, type 2 diabetes, and even cancer. But the missing link was not deciphered. And we asked ourselves whether maybe the circadian microbiome could at least partially explain this. So to answer this, we performed experiments in mice in which we subjected them to severe disturbance in their circadian activity by well-known models. We did this also in humans in a very cute experiment in which I took many of my postdocs and flew them all over the world and only asked them to collect their poop while they were jet-lagged before and after. We characterized their microbiome along the circadian disturbance and found that it was disturbed. But as I said at the beginning, we really value causality and mechanism, so we transferred the jet-lagged or non-jet-lagged microbiomes into germ-free mice, into sterile mice, and found, to our surprise, that the transfer of this circadianly disturbed microbiome induced a severe obesity and type 2 diabetes, both in mice and in humans. Meaning that at least some of this missing link may be driven by the microbiome, and we and others are now engaged in human clinical trials in seeing whether we can harness this time-resolved dietary behavior towards a microbiome alteration towards health. So all of these studies and many studies that I didn't mention, which focus on nutrition, the microbiome, and human health, are very interesting, also very promising, because they may enable to translate the findings towards improved human lifestyle and behavior. But in doing so, we were also tackled with the biggest challenge in this field, I think, the big elephant in the room, which is how do we measure what people eat? Now, if you think about it, the nutritional sciences have done this for decades, but basically, the only way we do this is by these food frequency questionnaires, these horrible, long, hard-to-follow questionnaires that generate very, very noisy data because they're different between countries. Sometimes the food components, even when they're identical, are called in different names. People forget, people lie, people are ashamed. So this generates very crappy data, and when the input is so bad, then you can't expect the output, even with AI, to be much better. So you may tell me, we have the smartphones, we can now quantify diet. There are many promising advances there, but I would argue, and we've just reviewed this, that in some discrete cases of foods that can be identified based on their components on the left, this could be a wonderful solution, but the more complex food becomes, the less you can disentangle the components, even by these digital methods. So this is a huge unmet need, which leads me to a completely unrelated story we were tackling and going after for 7 years. After another unmet need, which is our wish to develop a way to directly quantify microbiome function, not just by looking at the DNA level, which at best provides a snapshot of functional potential, because not all genes in the DNA get transcribed and translated, but actually to look at functions for the first time. And for that, you need to look at proteins. Proteins are one of the most important effector molecule in life. And people have tried to perform metaproteomics of the microbiome before us. The biggest challenge there is that you have a huge universal protein reference database with lots of overlaps. So the resolution becomes very, very poor. And what we have done after many years of miserably failing is to develop a little trick which involves the sequencing by DNA of a given, let's say, fecal sample, and the generation of a pair experiment or pair sample reference database, which is then the same sample is put through mass spec. So you have proteins from a given sample that are projected on a reference database that is generated from the same sample. Since it's much smaller and much more compatible, the resolution becomes almost perfect, the running time becomes shorter, it's less expensive. We were very happy, and we were able for the first time to allocate all proteins in a given sample to their species and strain origin, and basically identifying the functional microbiome. For the same price, the mass spec also provided the host responsive landscape in proteins to the microbiome, because the mass spec doesn't care which proteins it characterizes. It's both the gut and the microbiome. And using this system, we were able to generate a functional map of both host and microbiome interactions, and this enabled us to discover new biomarkers and therapeutic targets. This is worth a different talk altogether. But the point I'm making here is that in doing all of this work, we were able to generate an annotation of 97% of all proteins. 3 proteins didn't annotate to the host and to the microbiome, which really bugged us. So my postdocs, who are much smarter than me, came up with an hypothesis that maybe these small amounts of proteins actually come from diet. Maybe they represent dietary proteins that make it into the fecal sample. So to try and address this hypothesis, we generated a third reference database consisting of many hundreds of foods consumed in a developed country in this case, and the unique proteins that signify each of these foods. And when we projected the proteins on the third reference database, indeed, all of the proteins came from food, which for us was a eureka moment, because it represented an opportunity that maybe we can start quantifying what people eat, the dietary exposome, objectively based on fecal samples. Of course, the burden of proof was on us. So I started at the population level, taking advantage of the fact that I had 2 entities, one in Israel and one in Germany. So we've allocated 2 healthy cohorts of individuals from both countries. We collected their feces. We performed this metagenome-informed metaproteomics on them and looked at the dietary signals. Now, for example, when we looked at wheat signals, we found that the signal was comparable between the 2 populations. You know, Germans eat pretzels, and we in Israel, we eat pitas, but wheat is wheat, so the results were very comparable. But then we looked at, for example, pork. And when we looked at pork, we saw a huge German signal and a 0 Israeli signal. And this, of course, stems from the fact that all of the participants in the Israeli cohorts were either Muslims or Jews, and we just don't do pork, giving us a hint that maybe this method was actually catching the right signal. What was really exciting for us was that, for example, the pork signal didn't just stem from a single pork unique protein. There is a number of unique pork proteins that all behave the same. So the signal is very accurate, very crisp, and very trustworthy. To compensate for the loss of consumption of pork, the population in Israel eat much more chicken, and that could also be caught using this method. So in the population level, this method seemed to be working quite nicely. And then we moved to the personalized level. So I'll give you a couple of examples of trials that we've done. We took a group of healthy individuals, kindly asked them not to consume 4 foods, bananas, apples, peanuts, and cucumbers for a few weeks. And we gave them pairs of these foods during predefined days, and we performed this methodology that I've just described consecutively on different days during the trial. And indeed, we could see that the right foods that were consumed on predefined days were spiking at the right time, meaning that, again, this method could catch, even at an individualized level, the consumption of different foods. And again, each of the foods was defined by multiple unique proteins, making the signal very trustworthy. Then reviewer number 2 kindly asked us to look into the sensitivity of the method. So we performed another clinical trial in which we gave healthy volunteers either 5 peanuts a day, then washed out, then 20 peanuts a day, then washed out, then 40 peanuts a day. And we measured their peanut signals in stool, and we could generate a very nice dose-response curve. And then you can go to disease settings. For example, we looked into children with new onset celiac that were just diagnosed. And you can see that when they came to the doctor, they were all eating bread. The signal was high in all of them. They then changed into the first-line treatment in celiac, which is a gluten-free diet. And we followed them, and you can see that the signal goes to the floor in all of the children, other than a single children. And then we went back to the child, and we found that the child was cheating. So collectively, we call this method IFOMED, Integrated Proteogenomics of Host Microbiome Diet. It enables to decode the 3 major partners that modulate disease in the gut: the host, the diet, and the microbiome. We compare them, we benchmark them against the gold standard food frequency questionnaires. The agreement is around 90%. We feel that the 10% is in our favor, but we cannot prove it against the gold standard. And just as a last slide, you can already harness this into disease. So for example, if you take a group of pediatric Crohn's disease kids with Crohn's disease, their first-line treatment is a dietary intervention consisting of elemental diet supplementation, meaning that you give them a part of the diet that is composed of discrete amino acids. So you would expect that our signal would be reduced because the mass spec cannot identify discrete amino acids. And indeed, this was the case. And actually, the slope of the reduction in the signal was corresponding to the compliance of the kids to the dietary treatment that they were receiving. And what was cute is that in the same sample, for the same price, we can also quantify the signal from the microbiome and the signal from the host, and we could correlate the inflammatory signal from the host based on many different inflammatory proteins to the level of compliance. So you can already tell whether a child is compliant and what this does to its intestinal inflammation. So to summarize, I've introduced in this last part IFOMED, Integrated Proteogenomics of Host Microbiome and Diet, that enables us to reach a better holistic and functional understanding of host microbiome interactions and the diet. And of course, this can be applied to any aging-related disease or non-aging-related disease. From a translational standpoint, and I've not had time to show you, this enables to identify new trans-kingdom biomarkers that outperform the current biomarkers used clinically. Quantify diet and digestion, therapeutic targets can be assessed, and you can go back to personalized nutrition from where I started the talk and integrate these measurements into the machine learning and AI computational approaches in order to make them more accurate, which is what we're doing as we speak. And I'll finish by thanking the many members of my 2 labs at the Weizmann Institute and at the German Cancer Research Center in Heidelberg, who are doing very ambitious and very amazing work. I'm very grateful for them. The many, many clinical and scientific partners in all of these adventures, there are too many of them to put on one slide, and the funding agencies, and I think I'll stop here.
Thank you, Jeroen. This was a wonderful presentation and a very good way to start the symposium. In the interest of time, I'll take one question. Maybe it's going to be my question then, unless, Jens, are you coming for a question or no? Okay. Oh, there's a question there. Yes, please.
Thank you for your great talk. I'm doing metaproteomics in Korea, and we are trying to find out what determines the immunotherapy response. But I actually consider about the diet influence in immunotherapy response or not. So did you find any cues for dietary signal can determine the immunotherapy response, like it makes inflammation or suppress the immune response?
Yeah, very good question. And as I said, we think that this platform could help advance the field towards direct, quantifiable functional assessment of all three major partners. In this work, we've started by mainly focusing on inflammatory bowel disease, being another hallmark disease that is impacted by host genetics, by dietary factors, and the microbiome. And we've gone a long way forward with that. And as you can imagine, we're now doing this with many collaborators in many other diseases, including in cancer, mainly in my German cancer center. So the jury is out there, but I think, given all the previous insights on microbiome involvement in immunotherapy responsiveness, I think we could expect interesting and maybe surprising findings there.
Thank you.
Thank you very much.
Thank you, Andre, for this lovely introduction. Thank you for the organizers, for the Danone Institute for inviting me. So I'm talking about dietary modulation, and I have disclosures today because I am a co-founder of Symbiotic Health, and I will show a few slides of their probiotic work, so I would like to declare this. So I wasn't aware that I was working on aging, so I was very surprised I got invited. But I'm definitely interested in how the microbiome interacts with these chronic diseases that have already been mentioned several times, and how this is then shaped by diet. And since many of these chronic diseases are linked to older age, and giving also the fact that we should be probably eating healthy all through our life to prevent these, I probably have been working on healthy aging all around and all along. So this is why I then really enjoyed actually presenting this presentation. I'm also surrounded in Cork by absolute experts. So this is Paul O'Toole, a grumpy Irishman, and he is one of the world leaders in this area of microbiome and aging, has published very, very important papers showing that we have a dysbiosis, for example, the older we get, especially in not so healthy aging, where we have pathobionts going up, protective taxa going down. We also see inflammation, or how some scientists then refer to this as inflamm aging going up, often related to a disruption in barrier function. And then this is linked then to other readouts of aging, such as frailty and other cognitive struggles. Yeah. This is one of the classic papers. This is a slide Marcus Claesen provided me, who is also a professor in Cork. This is their famous paper on the elderly, published in Nature in 2012. Really interesting work, showing one of the first studies showing how diversity actually declines in older age. And very, very interesting then ecological findings. For example, if you plot their fecal microbiomes, you can see then the elderly actually cluster pretty much with younger controls as long as they still live in the community. And when they then move more into care homes, their microbiome really degrades. It goes down in diversity. Obviously, there's a lot of ecological factors that then contributes to this, but they also already did preliminary analysis linking this then to the unhealthier diet that is served in these Irish care homes. Yeah. And that obviously then already, made them suggest, there is a huge, rationale to modulate with diet the microbiome to support healthier aging. And this is my outline today, so I'm going to show you some evidence that the microbiome is in fact linked to longevity and aging. And because it's very hard to establish cause and effect from human studies, I'm going to show you some evidence from rodent models. I'm going to show you some evidence from the nutrition field, how eating is linked to healthy eating. Then I'm going to show you 2 human studies that are, I thought, intervention studies on diet that I thought are of relevance as it relates to aging. Show you some of the microbial metabolic mediators that are likely to link diet to healthy aging. And finally, look then beyond, diet and for other microbiome-centered treatments and a potential role for bifidobacteria. So one of the few advantages of advanced age is that you know the older literature, and this is one of the and there is a lot of fascinating work on animal models as it relates to aging. So this is a publication from 1966 looking at comparing germ-free versus conventional mice, and very much surprisingly, these are the mice with a microbiota, and here you can see the germ-free counterparts, and as you can see, there is a substantial elongation of lifespan in germ-free mice, and the same work has been replicated also in rats. Also, at least in this dietary context here, the microbiome actually shortens lifespan. A study then later on confirmed this finding here, that the germ-frees actually live longer, but what it also showed is that this is on an adequate diet. If you dietary restrict these mice, then actually the negative effect of the microbiome goes away and even becomes slightly beneficial. And you can also say dietary restriction is even more important than actually the presence of a microbiome. But there's definitely an interaction between diet and the microbiome as it relates to lifespan. And then more recently, some really nice mechanistic work that was published in the last 10 years. These are 2 studies using mice and causal experiments in which actually older microbiomes are transferred to germ-free mice or through co-housing. These experiments actually confirmed much of this older literature that the microbiome is really often costly and that germ-free counterparts live often longer. They established causality. The microbiome is really causally related. Very elegant work here with transfers or co-housing. And finally, they define some of the mechanisms that I've already introduced, that you have a dysbiosis because older microbiomes can be transferred. This is then linked to reduced barrier integrity. You have more bacterial translocation, and then you have this systemic inflammation that is then linked, to aging phenotypes. So very, very interesting, causal, foundations of this exact field. And I also think this raises some evolutionary questions about, how is the microbiome actually, what is our relationship with the microbiome as it relates to aging? Because obviously the microbiome was important in evolution. We have clear contribution to fitness, but this is obviously during reproductive age, and during as we get older, this seems from these animal models that the microbiome might become costly, and there are some detrimental effects due to barrier decline, bacterial translocation, and then inflammations. And this could, of course, now just be an evolutionary trade-off, meaning the microbiome has evolved to support fitness during reproductive years, but it also, in evolution, it could also be antagonistic pleiotrophy, meaning that it actually evolved to shorten lifespan, and by this actually increase the fitness of the population. And the first time I read this in a publication was here in a paper authored by Ruth Ley, who I'm really, really pleased to be on the podium. And this was really just one of the many times where a publication of Ruth completely blew my mind. So the microbiome might actually have evolved to kill us off when we are getting older to then, generate space, for the younger offspring. That's a hypothesis, of course. No matter if that's true or if it's just a trade-off, it's definitely that there can be a cost associated, and these animal experiments showed this is diet dependent and again provides a rationale to look at diet. So how should we eat to age healthy? So this is a very recent publication in Nature Medicine by the folks from Harvard School of Public Health. They looked at different, also at their big nurses studies and health studies, so hundreds of thousands looks how diet is linked, and the best indice actually that links with different, hallmark of aging, healthy aging, chronic diseases, survival past 70 years, cognitive health, you can see is this alternative healthy eating index, performs even better than some indices around Mediterranean diet, performs much better than actually a healthy plant diet indice, probably because there's a lot of not so good plant, processed foods. The reason why this is probably working so well is it's an US population, so it looks at fruits, whole grains, nuts, legumes, but it also specifically looks as detrimental, processed food, sugary drink, but also then processed meats, where there is very, very strong rationale that they interact actually with the microbiome. If you look at the individual foods, you can see the usual suspects here of all the beneficial fruit, whole grains, vegetables. Yogurt shows up very high up, so the Danone folks didn't tell me to mention this, but it is very good, and it also has a FDA claim on the prevention of type 2 diabetes. That's important, but what is more important is wine shows up here. So you can really elongate your life by moderate consumption of wine, unfortunately. And then what is probably just as important is all the negative stuff here, and red and processed meat show up, trans fats, so also the usual suspects, but again, in terms of the microbiome, this is probably very important. There's a second study that I thought was very interesting in a UK cohort, also showing that you can actually extend your life expectancy by 10 years. Again, the usual suspects, you can see very, very high consistency. And what is very interesting in these studies, in the models, is that they show you can extend your life expectancy when you start eating healthy in your 40s, and if you start eating healthy in your 70s, it's only around 4 to 5 years that you can add. Again, showing that you should probably start eating healthier a bit earlier on, but there's still, no matter when you start, you can add life expectancy on average. Yeah? So how does this relate to the microbiome? This is a paper we published, a review actually years earlier, but I can tell you there is not a single disagreement with these studies on what is proposed. You have the usual suspects here of the healthy foods, and probably very little surprise, fiber, short-chain fatty acid, phytochemicals, polyphenols. But then what is probably just as important are these negative aspects, the processed foods, the saturated fatty acids, and the then metabolic problems that arise from consuming, for example, high amounts of saturated fat, and I'm coming back to this in terms of bile acids, but also a higher proportion of protein, which is then fermented in the gut microbiome to detrimental outcomes. And again, high protein diets, again, in mouse models are actually linked to shorter life expectancy. So what do we know from intervention trials? This is, again, a study led by Paul O'Toole. This is, stands out. This is the New Age study. It stands out because it's a large intervention trial with a Mediterranean diet in 612 adults, several countries. They put them on 12 months with dietary advice, showing very nice that first, markers of inflammation and chronic disease and frailty go down. This is then linked to bacteria that are correlated with frailty and cognition. You see increases in short-chain fatty acids, reduction in secondary bile acids, also a reduction in fermentation output from proteolytic fermentation. This is independent of age, interestingly, but there's also some real issues with actually having the elderly stick to a diet like this. So there are definitely challenges around this detention here. Some of them don't have teeth anymore, and also some of them just don't really like the diet as much. Some of the data here, you can see we had a lot of talk at this conference about Faecalibacterium prausnitzii, Roseburia, other short-chain fatty acid producers. In here, for example, we have certain Collinsella, but we also have Ruminococcus torques, a known mucus degrader. We have then strong correlations which very much make sense with marker of inflammation, and then reverse or positive or negative dependent on which direction the bacteria go, but then also correlations which actually markers of aging, such as hand grip strength, for example, and that is pretty consistent amongst the different countries in which this diet has been performed. So very nice. Yeah. So I would like to show you some of our own study. So this is the study on the non-industrialized type diet that we published in Cell last year. This is not a study in an aging population. Actually, the average age was 25, but as I mentioned, should probably start eating healthy early on. But it's also relevant because it was in contrast to the study we've just mentioned, and Aaron mentioned that it's hard to control for diet. So this is a strictly controlled diet study in which we prepared the meals and actually gave them. And it's also interesting because it's a diet that mimics the dietary pattern in non-industrialized populations, which show often very low levels of these chronic age-related diseases. So this is a fascinating paper in Lancet showing that a tribe in Bolivia, the Siame, have very, very low levels of heart disease, very low level of plaque in their arteries. This is a publication from rural Papua New Guinea in a population with very little stroke and heart disease. And then again, in the Sami, there's evidence that dementia is actually very low. So it is worth to considering how these populations eat. This is not primarily driven by life expectancy, because life expectancy is short, and this is driven by infections, but if these individuals go older, they still have much, much lower levels of these chronic diseases. This is, again, this Lancet paper showing that there's substantially less plaque in this Bolivian tribe, 5 times actually low, again, and then resulting in much, much lower levels of actually heart disease. So how do they eat? Actually, it's not an extreme diet, but it is actually quite high in complex carbohydrates, low in protein, very low in saturated fat, and then very characteristic of these non-industrialized populations, very, very high in dietary fiber, more than we actually recommend in dietary guidelines. Of course, no industrialized foods and no added sugar. And so this is very similar to this non-industrialized type diet that we developed that was based on the eating patterns in rural Papua New Guinea. Lots of shout out goes to Anissa Ahmed, who is now a postdoc in the University of Alberta, who turned this dietary information into meals that are actually acceptable to Canadians to eat and to be tested in a human nutritional trial. So this is the characteristics, very much in line with how these Sami eat, but also in line with what has been linked to healthy aging, very high in dietary fiber, for example. And then thanks to a collaboration with Catherine Field and Carla Brado at the University of Alberta, we could do a strictly controlled nutrition trial where every single meal was handed to the participants. And poor Anissa, during her PhD, had to spend 1 year in a kitchen cooking meals for inhabitants and handing them actually out. Good thing is, it really shows very strong findings then. So these are the microbiome shift. Strongest is the genus Bifidobacterium, but then we see Faecalibacterium, Roseburia, similar to what Paul O'Toole saw with the Mediterranean diet. Bacteroides, for example, goes down. Bilophila, a pro-inflammatory bacterium, goes down. And here again, Colin Seller going down. So very consistent findings. If you look functionally, the fecal pH drops. And for me, this is exciting because I think this is a really underappreciated marker, the importance of actually the pH in the gut. And I was involved in this nice review here by Tina Rusk-Light, showing how many metabolic processes you can actually change by reducing the pH. One being, for example, the reduction of proteolytic fermentation, which we in fact showed in a reduction of branched-chain fatty acids. We also saw an increase of this metabolite indole 3-propionic acid, so that was in plasma. It's a microbial metabolite linked to a whole range of benefits. It's neuroprotective, for example. And then we also saw a reduction of secondary bile acids. And there have been already a few talks around secondary bile acids, and they're important in signaling. And I don't want to take any of this away, but these are also established carcinogens. And there's just a really nice paper by Tom Clavel and other individuals clearly causally showing that secondary bile acids are actually causing colon cancer. So I do think that this is, and again, also this is consistent with what Paul O'Toole showed in the Mediterranean diet study. Yeah. What happens to cardiometabolic risk markers? First, these individuals lost weight despite the fact that this was a calorie-controlled study. So you actually consume or less accessible calories at least. And you can see very, very strong effects. Fasting glucose goes down by 6%. These are healthy individuals, but LDL cholesterol goes down 17% in just 3 weeks when you go onto this diet, and CRP, mark of inflammation and heart disease, goes down 14%. So very, very strong effects on cardiometabolic risk markers. And we work with machine learning gurus like Dan Knight and Ross Kreiner showing that, and again, this is not establishing causality, but if you look into the metagenomics, actually one predictor or the dominant predictor of the reduction in CRP are actually mucin targeted to total metagenomic reads, at least suggesting that actually an improvement in barrier integrity might be responsible for this, anti-inflammatory effect. Yeah. So this, again, this wasn't a study on the elderly, but it probably still shows certain mechanisms by which we can modulate the microbiome to improve then healthy agings. And, I'm sure Sean is probably going to talk a little bit about this too. So some of the usual suspects, butyrate, for example, but then also I mentioned mucus degradation and also, the improvement of this barrier function and then anti-inflammatory effects. And obviously, this is complex diets. They're very reliable. We actually see very little variation in the responses, in the benefits in our study. But obviously, it is very, very difficult to establish whole diets like this then in an aging population. And this is really where we could think perhaps about some, as dangerous as it is, to look for magic bullets, but also for alternative therapeutic approaches to target this. And the one thing with symbiotic health we're interested in is bifidobacteria. And the reason is simply they are linked to health, and this is data produced by symbiotic health. And you can see there the dominant two species, this Bifidobacterium adolescens and Bifidobacterium longum. They're actually quite dominant in a healthy gut, but then decline actually the older. As I mentioned, they do respond to this NEMI diet, but they are also, can be targeted by prebiotic, and they can amplify other fiber effects through cross-feeding. And then there are a lot of, and then there is some really interesting links actually in elderly populations, and there's a lot of variation here amongst global populations. I don't want to simplify this too much, but this is a study here on centenarians in Sardinia, one of these populations that eat quite healthy, and you can see in the centenarians bifidobacterium, and specifically this Bifidobacterium adolescens, substantially higher than in just healthy elderly, for example. Yeah. And we have quite a bit of functional work now linking B. adolescens, for example, to the production of GABA. We see here that there is studies that it ameliorates sarcopenia, which is often associated in the elderly. This is our own human trial in adults showing that it does improve barrier function. And then finally, this is a nice paper here showing that in different models, it actually increases health span and lifespan that can be achieved through this Bifidobacterium adolescens. And with this, I'm out of time. So what the take-home messages is, I think the microbiome is a plausible contributor to aging. In animal models, we have clear evidence. In humans, it's always hard to establish this. Diet is likely a factor of this. To modulate the microbiome, things of just, for example, calorie restriction is already important, but then we have also diet like the Mediterranean diet or the NEMI diet showing that this can actually influence host microbe interactions that are relevant to aging. Here again, calorie restriction might be one mechanism because it is easier to actually restrict calorie with this, but then we have other healthy components of these diets. It's very difficult to implement this, specifically in the elderly and specifically once they are in care homes. But to improve healthy eating, you should really encourage to eat, already healthy earlier in life. And finally, there is scope for microbiome targeted intervention to improve health spans. But we need much more clinical trials, specifically also some that look at microbiome mediations. If you would think of how to do a trial like this and what the most important clinical outcome would be in a trial on healthy aging, this is me and Itzik Mizrahi in Seoul in 2014. I'm probably still not completely recovered from that week, but I can say we were in a nightclub 3 times in just 5 days, dancing all night. There's no way I could still do this, but I would be very keen to see a probiotic who can actually help me with that again. Yeah. So these are all the individuals that actually contributed to our NEMI study, would acknowledge all of them, also acknowledge the Papua New Guinean Institute and the people of Papua New Guinea, the funders. And with this, if you're interested in to eat a bit more non-industrial, all the recipes are freely available, and you can download the recipe book from the internet. So thank you for your attention.
Thank you, Jens. That was a great talk. I'm going to allow a few questions. While people are coming to the microphone, I may ask the first one. I was fascinated by the switch from short-chain fatty acid to branched-chain fatty acids with the Restore diet. And that brings into the question the role of branched-chain amino acids, because one of the best predictors of the risk for type 2 diabetes is actually the plasma level of branched-chain amino acids. So were those affected by the Restore diet, or do you think it's mechanisms that are independent from these branched-chain amino acids, and it's entirely gut microbiome dependent?
So unfortunately, I don't have an answer. Yeah, we didn't access them. They didn't show up in our metabolomics analysis, so we don't actually know to what degree they are affecting in a diet like this. Yeah.
Okay.
So, yeah. But you're right, they are one of the strongest links to actually type 2 diabetes, for example. Yeah.
Thank you.
Yeah.
Question. Amazing talk, really interesting. So I have, in a way, I have two questions that are completely separate. One is about what you said at the beginning, this theory that after not being procreative anymore, then the microbiome could kill us. But evolutionary, we're like, I mean, that's not your theory, but evolutionary, I guess, 1,000 years ago, we only kind of managed to get to 40-ish. So, but now we have this industrial diet, which is quite unhealthy, as you showed, and we still get much older. Maybe, do you have some context in your
I don't think we necessarily get older because of the industrial diet, you know, so okay, to some degree, because of course it provides security, food security, so we've completely, you know, stopped starvation and things, but once this is, I think the life expectancy is medicine, you know, and treatments and better and prevention of infections. And this is also when you actually look in these non-industrialized populations, their life expectancy is shorter. But if you actually once, you know, you are out of the period where you have infant mortality, then often they become actually quite older, you know. So I think that the evidence, that, at least from the observational studies, show that these healthy diets actually expand life expectancy. And I don't know if I read into your question, but clearly evolutionary, none of this probably played a role, you know, because as you said, you know, throughout human evolution, you know, we probably, you know, didn't get that old. But then the hypothesis is still, you know, that there might be a pleiotropic effect. It's a hypothesis. I like it, but I haven't tested it. I don't know if Ruth ever thought about testing it. Yeah, so, yeah. Yeah.
All right, and the other one was about the beefidos and the centenarians. I don't know how deep you dug into that, but are they different than the same species in healthy adults or even in kids?
Oh, that's a really good question. I actually wouldn't know that. Yeah, and I also have to say this is one population of centenarians where they are very dominant. There are other, and I don't know if Sean looked into some of, but this is quite confusing because there are even centenarians in China then which are dominated by bacteroides, for example, which we then in Western courts actually link more with detrimental effects. So it's one population, and it's, you know, it's one, we need the human trials, but it's just one potential strategy, you know, by which we perhaps can actually do interventions then. Yeah. Yeah.
Thanks a lot.
Thank you. Very, very short question.
Yeah, very quick.
Short answer, please.
Yeah, very beautiful talk. I'm curious about the composition of the diet you use in the trial, and I'm wondering, does that diet contain low animal protein compared to, you know, Western diet, control diet, you know, you use in the trial?
So we mimic this on what, you know, rural Papua New Guineans would eat. So it has animal protein. It's chicken, fish, and pork, because that's what they eat. And it's low levels. Yeah, so it's one small portion a day, but it's not vegetarian. Yeah. So, and it's all in the book, so you can download it. Yeah. So as we have the entire diet plan and recipes, they're all there. Yeah.
Sounds good. Yeah, thanks.
Thank you.
Thank you.
Thanks, Andre, for that really kind introduction, to the Danone Institute for the invitation, and I'm so honored to be in the company that I'm in with Aran and Jens. And they both gave great introduction to a lot of the stuff I'm going to talk about, so I can whip through some of my slides and save a little time. I don't really think I'm an aging researcher either. It's about 6 years ago, I guess, we started thinking about aging in the context of the microbiome. And when we were reading the literature, we kind of observed two major patterns that were out there. What is aging in the gut associated with? Well, one signature seemed to be kind of managing the decline. You want to just put the brakes on bad stuff that's happening as we age. And this is where a lot of the aging field is, kind of trying to slow things down or rejuvenate you back to some prior state. But there was also work showing, that in centenarians or maybe in very healthy older people, there might be differential patterns occurring. And so, for example, in centenarians, there was this observation that there's a steady decline in some of the core taxa that dominate us when we're younger, at least on average, and then a rise of the more subdominant taxa in the gut. And this work was driven by a really talented postdoc from a few years back, Tomasz Wolomansky. And the major signature that we found when we were looking at this large wellness cohort that we had access to called Aravel was this thing called uniqueness. We sort of invented this metric, honestly, just by kind of squinting at the data for long enough to kind of explain what it is. If you think about the beta diversity, how similar are two microbiome profiles to one another, how distant are they, or how similar are they? If you project them into some 2D space, and every point is a microbiome, and distance is sort of how differential the ecology is between the 2 points, your uniqueness score is your nearest neighbor distance in this space. So this red point has a higher nearest neighbor distance than this blue point. It's kind of on the fringe of the cloud. And oddly enough, during the course of the lifespan, it looked like what was going on is you get this steady rise in uniqueness over the decades of life. We didn't quite know exactly what this meant. The Aravel cohort, it was a consumer scientific wellness cohort where people were coached on personalized wellness. And we had access to the data, thousands of people with lots of dense multi-omics. And so this is like a generally healthy set of people. So that's a caveat here to think about. And we validated this in many, many other cohorts. So if you look across the world even, and other groups have done some validation here too, you indeed see this pattern of increasing beta diversity uniqueness as we get older. It can vary a little bit cohort to cohort, but generally we kind of see this happening. And weirdly, it's associated with things that are happening in the bloodstream. And so we kind of correlated this uniqueness score with circulating blood markers in these thousands of individuals. And the major signature we see are these, well, Jens mentioned proteolytic fermentation of microbes. You see these amino acid fermentation byproducts popping up in the bloodstream. Not necessarily the good guys, right? We often consider these things as bad, like p-creosol or phenylacetyglutamine. These are associated with like heart disease or kidney damage. Yet, they're also super enriched in the bloodstreams of centenarians. And in fact, if you kind of Google phenylacetyglutamine, it's been patented as a biomarker of centenarian status. It's way elevated in the bloodstreams of centenarians, but also in their children's bloodstreams. So it's even a marker of likelihood of achieving a long life. I don't know what's going on. This is still kind of a paradox for us. My suspicion is that when we get a lot older, increasing our protein intake might actually be protective against frailty or muscle loss, whereas maybe in early life, higher protein intake is not so great. I don't know. But getting back to the microbiome, one of the kind of taxonomic signatures of this uniqueness signature is a depletion of gram-negative anaerobes that tend to dominate our guts when we're young, like Bacteroides or Prevotella, depending on where you are in the world, and a rise in a lot of different butyrate-producing Clostridia, which are the kind of blue colors here. So we see a lot of these different genera from this class of butyrate producers that tend to be elevated in people who have higher levels of uniqueness. Kind of collapsing that down back into kind of a beta diversity space where different distance between points is kind of similarity in the ecological structure of your microbiome. We see that uniqueness is clustered up here in the elbow of this curve in these thousands of people. You can paint this by different taxa. So Bacteroides are enriched over in this arm, Prevotella down in this arm, and the crux here is enriched generally in these butyrate-producing Clostridia. You can see a very strong negative association between this uniqueness score and the dominance of Bacteroides in the guts of people. Okay, so then we next looked at a cohort of older folks. So Aravel spanned the whole lifespan, people from 18 years old to 99. We then looked at the Mr. Ross cohort, and this was we had a collaborator who had access to this cohort. It was recruited to look at osteoporosis in men, which was an understudied problem. So it's 1,000 older men between about 75 and 100 years old. And our uniqueness association with age was there, but it was actually weaker than what we saw in the Arabelle cohort. So we thought to ourselves, okay, maybe there is this partitioning going on. Maybe there are differential patterns of aging in the gut. So we found markers of, we found markers of health in this data set, so we could partition people based on their walking speed. So you can walk faster when you're older, you tend to be healthier. Self-reported health, whether you had high or low medication usage, or this life space score, which is a measure of kind of how much you get around in your life. And a composite healthy is you had to be healthy according to at least 3 of these other prior metrics to be composite healthy. But no matter how you sliced or diced it, if you were healthier, you tended to show a stronger association between uniqueness and getting older. And if you showed less good health, that association went away completely. So it seems to be this thing that's happening. There's differential signatures going on here. We also had follow-up data in this Mr. Ross cohort, so we could partition people based on their baseline microbiome composition and ask what happens with survival over a 4-year window. In the Kaplan-Meier here, you can clearly see that if you have higher Bacteroides dominance at your baseline time point, you're more likely to pass away at over 4 years. To control for other covariates, we did Cox proportional hazards, did a lot of different versions of it here, but in the kind of fully multivariate adjusted model, where we're adjusting for age, clinical site, body mass index, self-perceived health, and diagnosis of congestive heart failure, so trying to account for other factors that could be related to mortality. Controlling for those, if we look at people who've achieved very old age, 85 plus, we see very significant associations. So if you have low uniqueness or high Bacteroides, you're more likely to die over a 4-year window. So, it's correlated with health in older age, and it's also predictive of survival. Whether this is a causal signature or this is a survival effect is TBD, right? The older people might just be enriched for these signatures because they happen to survive. So we're still trying to understand what exactly is going on. We wrote a little perspective piece after this paper, kind of pushing back against some of the thinking in the field. So should we rejuvenate the gut, right? Should we give FMTs from 20-year-olds to 80-year-olds? Maybe not, is our take home. Because a lot of folks, when we're young, have this Bacteroides-dominated microbiota, and perhaps if we start transplanting that into very older folks, that might not be so good. And maybe it's just fine in a 20-year-old, but in an older person, it might actually do some damage. It seems that a decline in the dominance of these gram-negative anaerobes and the rise in these butyrate-producing Clostridia is associated with better outcomes later in life. We don't know what the mechanisms are driving these patterns. I'm sure it's diet. I would guess diet and lifestyle. And then how do we target? What is the outcome variable we want to target here? I don't want to live to 100 years old and be decrepit for the last 40 years of my life, right? I want health span, not lifespan. So let's think about what are metrics we want to improve with interventions that are focused on healthy aging. So then thinking about that, we moved to frailty metrics. So a really cool colleague joined my institute, Dr. Alice Kane. She was did her postdoc at Harvard in aging, and then came to ISB, and she looked at frailty in mice and in humans. And in this human study, she found that if you look at survival outcomes in a human population, frailty does a whole lot better at predicting survival than other things like metabolic syndrome, like chronic diseases, essentially. So it seems to be a very useful marker of outcomes, of health, and of survival. So we have the ability to now calculate these scores in the Arabelle cohort that I talked about earlier. So we can do this either through looking at clinical chemistries, so things like LDL cholesterol, A1C. We can aggregate a bunch of these things and calculate a score. We can also use self-reported questionnaire data about health and create a score there, or do a merged score. We tend to now use the merged score because it seems to be doing a little bit better job. Alex Carr in the group started running regressions on the microbiome data versus frailty. If you control for age, sex, BMI, and the first 4 principal components of genetic ancestry, this is the kind of stuff we see, which is kind of what we were expecting to see. So you pretty much get a lot of butyrate producers that are negatively associated with frailty, independent of age and these other covariates, and then a bunch of kind of a rogues gallery of bad guys that are positively associated. About 15% of the variance in frailty was explained by the microbiome after adjusting for these other covariates. So it's not nothing here. There's a fair amount of the variability in our healthy aging phenotype that seems to be explained by the microbiome. This also validates in other data sets. So this is a recent study in Nature Aging from another group in China, and they found the same exact things associating with frailty. It was essentially positive beneficial effects of butyrate producers and negative associations with a lot of the gram negatives, like the Bacteroidetes and the proteobacteria. So butyrate, I've been saying butyrate a lot. What is butyrate? It's a short-chain fatty acid. Jens brought this up before as well. It's probably the most potent of these short-chain fatty acids that the microbiota produce. It's super important. The only way we get it is through our microbes, and we're really underexposed to it in the developed world because we don't need as much fiber as we used to, right? We're lucky to get 10 grams of fiber in the US a day, whereas maybe we got 50 to 100 grams historically. So we're underexposed to this thing. What does it do? Lots of stuff, but maybe I'll point you towards one of its main functions that we know about is it's an HDAC inhibitor. And so, for example, it affects T cell differentiation. It biases T cells towards T regulatory cells rather than Th17 cells. And it tends to lower systemic inflammation across the body and help to normalize metabolism as well, like blood glucose and so on. So we want to make more of it. But it's hard to predict butyrate production from a given person's microbiome because whether you produce a lot of butyrate isn't simply a function of how much Faecalibacterium prausnitzii you have in your gut. It's a coincidence of both your dietary intake and the complex cross-feeding interactions that are happening in your microbiota. Our group has done some thinking about how to do this integration and do these predictions. We've built a tool called MyCom. Christian Diener, a very talented research scientist in the group, who's now faculty in Austria. It essentially is a tool that allows for flux balance analysis, genome scale metabolic modeling of complex multi-species communities that consist of hundreds of taxa. You use sequencing data to constrain which organisms are in the system. You pull down curated genome scale metabolic models from those organisms to make a kind of simulacrum of their gut metabolism, and then you constrain these models with dietary intake. There's a lot of ways to do that. We strip the diet of components that are consumed by the host, so things that are absorbed in the small intestine by using the Recon 3D human metabolic reconstruction, and then add a few things like mucin and bile acids to the milieu, and then we can predict growth rates of all the bugs in the system and the kind of fluxes of net consumption, net production, and the cross-feeding of metabolites in the system. We call these things microbial community scale metabolic models, or MCMMs. Okay, so we did a little bit of validation of these predictions in a prior paper by Nick Bowman. I'll just show you one piece of that analysis. We had data from Arabelle again with blood clinical chemistries, about 130 different things you can get measured at the doctor, and predictions of butyrate flux from their guts, making a very dumb assumption that they're all eating the standard American diet, which they're probably not all eating that, but close enough. And when we do this, we find that there's this many metabolites, 20-some metabolites that survive FDR correction, and they make sense with our intuition. So a lot of these are markers of lipid health. So if you are predicted to produce more butyrate in your gut, you tend to have lower LDL cholesterol, lower triglycerides, you have better insulin sensitivity, and you have lower systemic inflammation in general. So we think that it's telling us something resonant or useful or real biologically. What if we apply this to this uniqueness metric we were looking at before? Well, this is all kind of new work now that I'm going to show that hasn't been published yet. But when we run these models on the Arabelle population and look at uniqueness versus predicted butyrate, they're pretty well aligned actually. R of 0.53, not perfect, but it's pretty strongly associated. If you have higher levels of uniqueness, you are predicted to produce more butyrate, assuming a standard American diet. And this, aligns with this idea that the Clostridia are increasing as well in this group. So now I'm going to take another kind of hard pivot here towards statins, and this does actually make sense, and it relates to what I said before, and I'll try to explain. So we had another paper by Tom looking at how the microbiome's composition can affect our responsiveness to statins, this drug that lowers cholesterol in the body. And we looked at what are called enterotypes. This is a method of coarse graining or clustering microbiomes into groupings. We did this because another group had used this method on a statin study, and we had to kind of replicate some of their results. But it ended up being kind of useful. So this is, again, the Aravel PCOA that I've shown you before, this kind of V-shaped thing with this elbow and two arms. And you get 4 groups reproducibly. You see this in Europe and the US especially. There's a Prevotella group that's dominated in the genus Prevotella. You have this Ruminococcaceae group that's dominated by gram-positive anaerobes. And then you have 2 groups dominated by Bacteroides, BAC1 and BAC2. What differentiates them is this BAC2 group is very much depleted in Faecalibacterium, which is this dominant butyrate-producing microbe in the gut. And if we link this back to uniqueness, it's sort of, it's inverted, this plot, but it's the same people, essentially. And you see that it's the high uniqueness folks here that are in this Ruminococcaceae cluster, essentially. Okay, so if we look at so poor metabolic health and obesity, I forgot to mention, has been associated with this BAC2 enterotype. If we look at the statin responsiveness, just to give you a summary of what that paper found, is if you look across these enterotypes, we find that the on-target effect of the statin is maximized in the BAC2 group. So if you're in this enterotype, you tend to have the strongest on-target effect of statins. This HMG molecule, which is the upstream molecule, the enzyme that's being inhibited is HMG-CoA reductase, and HMG builds up if that enzyme's being inhibited. In the Ruminococcaceae group, you still see a significant uptick in statin users, but it's not quite as much. And here's the LDL lowering here. But one of the side effects that's underappreciated about statins is they actually kind of somewhat increase your risk of type 2 diabetes and perturb your insulin sensitivity. And it's the same BAC2 group that shows the biggest perturbation to their insulin resistance when they take statins. So they're most vulnerable to the side effect, but they also get the biggest on-target effect. Okay, so going back to frailty and the Aravel cohort, if we group people into these enterotypes again, we find that it's this BAC2 group that shows the highest average frailty values, and it's quite significant. And then concordantly, this Ruminococcaceae group, this gram-positive anaerobe-dominated group, it shows on average the lowest frailty values in this cohort. And this is a very big mix of people. It's people from 18 all the way up to 90 something. So it's very heterogeneous, but the patterns are quite significant. If we project this back into our uniqueness scores and our predicted butyrate, it aligns well with our expectations. The Ruminococcaceae group shows this highest level of uniqueness on average. BAC2 is fairly low. And then predicted butyrate, as you might expect, Ruminococcaceae is highest, and then the BAC2 group is lowest. And we're stratifying by sex as well to kind of show you how that falls out. And this is work, I should say, by Alex Carr in the lab, postdoc. So one unique thing about the Aravel cohort is it's longitudinal. They actually had an intervention, so people were given a healthy lifestyle intervention. They were coached to improve their diet. That was mostly just being told to eat more fruits and veggies, and they were coached to exercise more. And so across this population, they were all called once a week by their coaches and pushed to do all this. And what's strange about this cohort, if you look at most human cohorts and do frailty metrics, you find that on average we increase in frailty as we get older. In Aravel, over a 6 to 12-month window, most people went down in their frailty. So the dashed line is your 0 line, normalized to yourself at baseline. So most people are improving in their frailty over time. But some of these enterotypes can do better than others. So if you're BAC2 and you're a male, you can really bring down your frailty. These are the folks that probably had the worst lifestyles to begin with. This enterotype, like I was mentioning, is associated with obesity, diabetes, metabolic syndrome. So if you improve your diet and you already have that lifestyle, maybe you just have more to gain from having a lifestyle intervention. And the microbiome allows you then to target people for these interventions. For women, it was Prevotella, the Prevotella enterotype that showed the biggest drop in frailty. We're still working on this analysis, but we do see mediation. We see some interesting hits. We often are seeing sex hormones pop up in a lot of different human phenotypes, and this is true here as well for aging. So if we look at your enterotype status as the treatment, which is a little weird, I know, for mediation, and then your blood, this hormone, what is it called, cortisone glucuronide, as the mediator on frailty, we do see a significant mediation. About 21% of the effect of the enterotype on frailty is seemingly mediated via the reduction in this hormone that's a derivative of cortisol in the Rumina-Caucasia group. This all validates in the Mr. Oz cohort. So if you look at a different human cohort, we also see this association with frailty. And if we change our Kaplan-Meier, instead of looking at Bacteroides dominance, but just look at the two enterotype groups, we see kind of the same thing we would expect. If you're enriched in the gram positives, you tend to survive. And enriched in the gram negatives, you tend to not as well. So the take-home points. I would plead with you, don't give fecal transplants from 20-year-olds to 80-year-olds. It's not like the blood boy thing that the billionaires are trying to do. Probably not a good idea. There probably are some 20-year-olds who are perfectly fine donors for an 80-year-old, but you got to find which one. There is no average microbiome, right? The average human has one testicle and one ovary. The average microbiome isn't a thing. I think we want to target these interventions. It looks like having higher dominance of these gram-negative anaerobes is probably not great for your health span. Higher levels of the gram-positive seems to be better. I'm pretty sure that diet and lifestyle are the things that are driving all of this, and we don't quite know what's going on yet. If anybody's out there reviewing grants for the NIH, we put one in, and it's going to be reviewed in July. We would love to dive into this. Like Iran, we have some methods for trying to infer diet from metagenomic data, and we want to try to see if we can learn out what are the drivers of these effects. And with that, thanks to the trainees who did all this work, the funders, and to all of you for listening.
Thank you, Sean. We have time for a few questions. Yes.
Yeah, thank you for this very interesting talk. I am curious about the arrival cohort and the intervention when you have a different frailty difference for some enterotypes. Do you have the enterotypes data after the intervention to see if participants changed?
That's a great question. We maximized our N by only using baseline microbiomes. So we have, for about half of the people, we have follow-up microbiome data. And we will look at this, yes. But I wonder, right? So what I suspect is going on is that having this BAC2 enterotype, you're just more likely to die. And especially when you get into your 80s and 90s, you just stop seeing those people in the population. So it's not that like the microbiome is evolving and changing per se, it's just a survival effect. But if we do see that you can transition from BAC2 to one of these other enterotypes, maybe there's hope for those of us who have another enterotype that we can get to the right track.
Thank you.
Claire?
Hi, Sean. That was great. Some of the latest things that I find fascinating from aging research is that it's not really a linear decline, but it happens where it appears to happen in physiological distinct bumps or phases, with the first one in the mid-40s, then the next one in the 60s, et cetera. Most of the stuff that I've seen linking microbiome with aging may not take this physiology into account. They may model age linearly. Have you looked at the possible associations between decline or changes in the microbiome during these specific phases of aging?
Yeah, I mean, this is something we called out in our original paper, because if you look at the uniqueness over the decades, it's pretty flat until you get to about 40 or 50, and then it starts to go up. And so that's like the first inflection point you were talking about. And then it's not a linear increase. It's like an exponential increase as you progress through the decades after that. So I think it does align very well with the immunology aging data.
Yes.
Sasha Zhernakova-Groningen, great talk, thank you. I'm curious about this uniqueness parameter. It's known and very well shown that other omics layers show the same, particular methylation, but also proteomics and even gene expression. I was curious if you ever compared whether microbiome uniqueness goes along with uniqueness in other biological measurements? Do people become unique in all aspects, or is kind of microbiome separated from other omics?
That's a great question. It's like this entropy theory of aging, right? Are we just kind of like losing regulation and drifting as we get older? And I think what's going on in the gut, it's more of a factor that we harbor a lot of different disparate species and genera from the gram-positive group, and we have like a fewer set from the gram-negative group. And then that, as the gram-negatives are declining, that drives this increase in uniqueness. So I think there's more coherence, I will say, to the microbiome signature, whereas in the methylation, it's just kind of a loss of regulation.
Or maybe a lot of effects.
Yeah.
As well from microbiome. I think it's very interesting.
Yeah, good question.
We'll take one last question.
Hi, really interesting talk. Thank you so much. I was really interested that you saw a difference in the enterotype that was driving the improvement in frailty between the genders. I was just wondering if you have any theories about this, or have you looked a little bit more into why that could be?
It's a mystery. So Alex Kane, who I called out, she specializes in sex differences with aging, and it's really interesting, right? In humans, women have higher frailty, but they live longer lives. Men drop dead earlier, but have lower frailty. And how do you square that circle? We see both in mice and in humans these really variable effects. We have one mouse study that I didn't talk about where we were using NAD supplements, which are very controversial now because maybe they aren't going down with age, or they don't look like they are actually. But when we supplement, we don't really see much direct effect of those supplements on host physiology, but we see an effect on the microbiome, and it's an increase in Anerotruncus colihominis, which is a butyrate producer that is enriched in centenarians, or it has been shown to be in a couple of papers. And it has an effect, it mediates lower frailty in one sex, but not the other in mice. And so the microbiome effects in sex seem to be intersecting, and they have variable effects, and I don't think we understand why yet, but I think it's really fascinating.
Okay, well, cool stuff. Thank you so much.
Great, thank you. So I did prepare a few questions, and I got some help from from the speakers and the participants of this roundtable. So let me just crack one question at you guys and see where it's going to lead us. So one question is, when we look at your presentation and of course we think about this, I think one question that pops in our head is, are there actually microbiome signatures of healthy versus unhealthy aging? It's a simple question, but I'm not sure we get the simple answer to that. So who would like to start addressing this question? Sean?
I can, yeah. So I thought about this a lot in the early days because we know the work from Cork, was focused on a lot of these assisted living facilities for a while, right? So you saw that people in long-term care facilities, it looks like they're managing the decline side of aging. It looks unhealthy. Things aren't going so well for them. They actually have higher Bacteroides dominance, for example. But then in that same Claassen paper, right, they show if you're community dwelling, then that signature sort of is displaced. And so you can kind of hold on to health better when you're not eating a poor diet, perhaps, or you're around a lot of old people who have C. diff infections and are getting you sick and so on. So I think there are different signatures. What I'm currently struggling with is whether this is a survival effect and how much of it can be changed. And I suspect it can be changed because people going into these long-term care facilities seem to be pushed towards this bad phenotype, although maybe they're just being enriched because they're already sick and they're getting pulled into that system. We have a little bit of data on individual longitudinal stuff where if you look at a person over time, we do see that with healthy lifestyle intervention, you see a little bit of an increase in uniqueness, but we only have like a 1-year time series, and it's not quite enough to have a really strong p-value. So I'm hopeful that you can push yourself in a good direction, but I'm not sure.
Maybe I can continue that.
Please.
I think definitely also believe there's a microbiome synchronous in healthy, unhealthy, healthy aging. But for instance, based on a large scale human cohort study, and we see microbiome synchronous are shared across different chronic disorders. So no matter which diseases you have, but actually common synchronous. And these synchronous were not explained by disease comorbidity. So therefore, I really think could be driven by aging-related biological process. However, of course, it's descriptive, right? I could consider the biomarker as you described that the uniqueness or higher richness, and also shared disease composition between healthy and unhealthy conditions. And doesn't really have to say it's really causality yet, not very hard to establish in human cohorts. Second thing, I think synchronous will probably not only see on the compositional level, and also, as I mentioned yesterday, on the genetics level, that we see massive association associated with age. And also very striking observation we found, even opposite way, seems aged microbes seems to have a higher enhanced capacity and functionality to promote good health, because they adapted to the declined physical condition, try to be even more effective. So a little bit opposite to our classical thinking about the microbiome role in aging.
And I could probably add too, again, I'm not involved in this research, but my Cork folks, they also work on signatures and have put lots of effort into it, and they are very focused on keystone species, actually, as an ecological approach where they say there are, Paul O'Toole would say there are 7 species that are kind of at the center of knots that are then connected, and they are having paper to link. Then what is cause and effect there, again, you know, is clearly not established. Yeah.
Iran?
I agree with all the previous comments. I think 2 important points from my perspective. One, as was mentioned before, in any case in which a correlation or an association was made between microbiome composition and a health or disease status, I think it's clear that the majority are passenger effects, secondary effects. So in the aging process, you know, you have ambulation, exposure to drugs, comorbidities, blah, 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 that probably account for many of these changes, but we're all interested in finding the minority of changes that actually drive disease or aging-related processes, and that's easier said than done, but we'd be very excited to explore. The second point is that we're also finding that some of the variability between studies and within individuals could be deconvoluted by looking more carefully on function rather than composition. So imagine a healthy centenarian in Japan or in the US, you would likely find a different healthy configuration in both populations, but if you start looking at function, it generates a better common denominator to health and disease features. And so I think the more this field will move away from naming bugs and compositions, but by incorporating functions, we would find which functions are present or missed in a given health context, and that would generate maybe more actionable results.
So to jump on this a little bit, to understand function better, because I think we all agree we have to move beyond just looking at numbers of the type of bacteria, but what they do, how they function. And most of you have talked today about using different omics tools, like looking at the proteome, for example. Some would look at the metabolome. So is there something you think, I think, should we look at the multi-omics perspective, or should we focus on one type of signature, which is more functional than composition?
I definitely think multiple omics are very necessary. As we heard a beautiful story from the metaproteomics data, and also metabolomics, as also Sean have mentioned already. I think this is pretty key because, as I said, signals were not easily visible only one level of information, only combined it, right? So we had genomic levels that we already probably extensively studied in the past decades. Now metapromics and probably also metabolomics starting rising because a lot of omics data we did on the host side. As a limited actually efforts still behind also in the microbiome side, and how to integrate the host human side and the microbiome side together, I think is another level thing. That Sean mentioned that the genome scale or metagenome scale metabolic modeling, and here maybe we should consider at the whole genome level of metabolic modeling or proteomic network essentially to has a close link between human and also microorganisms. Maybe even consider other non-bacteria components as well, fungi, virus, all together. Right, so this is more comprehensive. I think we get more closer to the true picture.
Yeah, good point. I think we focus a lot on bacteria for several reasons, but what about other organisms? What role they play into, aging trajectories? Should we look more at the virome? Should we look? So, Ruth, you want to?
Well, is this working? Yeah, I think in that sense, I think we all agree that we need longitudinal studies on people, especially if we're looking at aging with respect to younger people. You're looking at people with vastly different histories, colonization histories, might have had H. pylori in the past, this kind of thing, and other organisms too. I mean, from eukaryotes to just different exposures in the past, and that's really difficult to account for. And I think until we have good longitudinal cohorts on aging, it's going to be quite challenging to tease some of these things apart.
Sean, you wanted to add something?
Well, just kind of the multi-omic and like what else are we not measuring context? I mean, anytime you have an interface between two or three things, you're going to find new stuff. I mean, most people have thrown the kitchen sink of omics at most phenotypes, and they've done the low-hanging fruit, which is the individual omic analysis. But finding clever ways to thread together two omics you're always going to find cool stuff there. So I think people should focus on that. And then one of the things that we aren't measuring enough of, brought up by Iran, is diet. So, whether you have a microbiome composition that can do one thing or another is largely determined by whether you ate an avocado that day or an artichoke. And so the combinatorics of having microbiota knowledge and that dietary exposure knowledge and the phenotype data, I think that's going to open up a lot of interesting avenues.
After nutrition or food, and a lot of you have talked about that today, it's always fascinating to see our drugs. When we talk about the exposomes, a lot of the drugs we take are having an effect, a major effect on the microbiome. And of course, if we talk about aging and older populations, there's more and more drugs, right? That probably becomes even a bigger determinant of the microbiome. So how much do you think it's like two questions in one. So how much do you think this should be covered better, or should we look at this more carefully, and what tools should be developed? I mean, we talk about personalized nutrition. Should we talk also about personalized medicine for microbiome and its impact on aging?
Yeah, I think it's an exceedingly important topic, and it's bilateral. It's not just the impact of drugs on the microbiome, which could have downstream health ramifications. Equally important and interesting is the effect of the microbiome on drug metabolism, and I think there were quite amazing studies suggesting that 25% or more of all drugs are metabolized to one way or another by the microbiome. And I think from a bird's eye view, as a clinician in my previous life, I think that if you look on any drug that is given to humans, the most effective drugs always impact a minority of humans. So the best of the best antihypertensives, would impact 20 or 30%, and we kind of use this knowledge in order to mix and match medications and hope that this trial and error would hit the right person. But I think that revisiting this observation and trying to explain why 70 or 50% of a given population do not respond to a given drug could eventually involve the microbiome. So if we do the same kind of personalized nutrition thought process and apply it to drugs, I think we could maybe better harness drugs to the individual.
Sorry, may I add something to that? And definitely I agree with Eran already said, but a lot of area has been overlooked, actually diet, drug interactions. Because while we take drugs, we also get other dietary advice when you should take drugs. That actually, and so this is important for the microbiome or in the future healthy aging is really for the prevention, right? First, the prevention to prevent the development of disease. Also, the secondary prevention to prevent further second comorbidities. So then, how to also take diet at the beginning before you have a disease to help personalized nutrition, and then later to combine personalized nutrition with personalized medicine to also prevent the secondary prevention strategies and also improve the drug efficacy and the drug toxicity. I think this is also area we should also take diet, medicine interaction into consideration.
Sean?
Maybe to add a little quantitative flair here, because I have it on the top of my mind from the statin paper. The only reason we looked at statins is because we had this large cohort, and if you just happen to sample a bunch of people in the US, 20% of them are taking statins. So it was just a very common drug, and we had the power to look at this question. I suspect if you looked at any other drug, you're going to see these effect modifications. But for statins, we could explain something like 22% of the variance in the on-target effects in the whole data set. 10% of that variance was explained by age, sex, and body mass index. You could explain about 4 or 5% of the variance with the 9 genomic markers that have been identified from GWAS to be most associated with statin responsiveness. So the genome could explain, 4% of the variance, and then the microbes were explaining something like, 6, 7, 8% of the variance. So it's on par with the genome. So pharmacomicrobiomics, should be on par with pharmacogenomics.
So what do we do for these people that would benefit from statins because they have, lipid profile, which is pretty bad? And so do we remove them from statin, or are we trying to use a biotic approach in order to change their microbiome so that you don't get the side effects of the drug? Is that possible? Is it something that we may think it's possible very soon?
Yeah, there's actually follow-up work to our paper from China. There's a group, it's Shi et al., I think it's Cell Metabolism, where they put new statin users on the drug, and they looked at things associated with the response. They found that statins were reducing the abundance of certain Clostridia that were metabolizing bile acids, and there was a decline in ursodeoxycholic acid. There just happens to be a drug called ursodiol, which is ursodeoxycholic acid. So they gave a small cohort of statin users, I think it was only like 5 to 10 people. They paired the statin with ursodeoxycholic acid, and the ursodiol completely rescued the metabolic phenotype. So their insulin sensitivity improved, and they still had the same LDL lowering effect. So I think, yes, you can.
But if they'd been on Jens's diet the whole time, they wouldn't have needed any of this, or?
Yeah. Maybe ursodeoxycholic acid is mediating that effect.
That's why the book is freely available.
So we're moving to interventions now. So I think we saw a good example with Jens talk about the Restore diet as a way to really change the microbiome for the better, and for not only to improve health but improve I would say health span, as you mentioned. So now those that wants to do trials, those that are clinicians in the room, for example, would like to design some trials to test nutritional or biotic interventions in aging populations. So 2 questions. Should they do a study with people that are 40, 50, 60, 70? When should we start doing these interventions? And what should be the design of those studies in order to make sure we get the most out of these research, these trials? May I start with you, Jens?
As I showed this observational study from the UK cohort, when they looked into this, And their model said, you know, if you start eating healthy in your 40s, it's 10 years you can add, and if you start in your 70s, it's 4 to 5 years, you know. So I guess it's never too late, but I guess the earlier you start, you know, the better it is, and then it really just depends on what you try to achieve with your trial, yeah. In terms of how to design it, even that's a world in itself, you know. That's the nutrition. We in the group, we are very. Aaron mentioned how hard it is, you know, to make people eat a certain way or to even just measure it. So we just try to get away from this as much as possible and just we try to control. We don't do the Kevin Hall. We don't put them in prison, you know, for 4 weeks. But we do hand them every single meal, which is a really good experimental design because so there's actually no incentive really to cheat because, you know, they would have to spend extra money. They get the food for free. They save time. And from what we see, we have very little indication that compliance is not very hard. But then there are any other experimental design, you know, you can advise them. You can just look into a cohort. You can try to measure it. Like, so it's hundreds of different ways of doing it, yeah. But we personally, as in our group, we try to control as much as possible. But that comes then with the disadvantage that they are shorter, you know, because you can't do this for longer than 2 months, for example. So if you would want to go a year or two, you know, then you would need a different experimental design, yeah.
So if we go from diets now to biotics, so what should be the approach? I'm going to take the side of the prebiotics, just for the sake of discussion and debate. So with prebiotics, at least you can design the prebiotic to act on a niche or a very specific organism that you know will be beneficial. There will be one, probably 10 or 20 microbes that will benefit from it. So the chance of success is maybe greater than for a probiotic, for example, or even a cocktail of probiotics. Do you agree with me, or you think both of them have their chance to have an impact? Who wants to take this one?
Well, I respectfully disagree.
Okay.
I mean, I agree and disagree. I think you probably did this for the sake of question, but the reality is much more complex than just comparing one to the other. And I'm not going to start dwelling on it, but even if you look at the most simple prebiotic fiber-based intervention, I would argue, and I'm arguing based on data that we've generated, that not all fibers are created equal. There is huge divergence in the chemistry of different fibers. So expecting that a prebiotic would do something very uniform, given this divergent chemistry and the different effects on so many species, I think is maybe romantic, but less evidence-based. And we need to do much more work in order to refine what we know about these effects. With probiotics, don't start me. Let the others talk.
Okay.
Yeah.
So I can add to this. So we actually, when in the group, when we did the non-industrialized type diet, we had a parallel study on fiber supplements, and it's pretty in agreement. So while we had very, very low inter-individual variation with the complex diet, probably because, you know, they eat all kind of different foods, we had very, very large inter-individual variation with the single fiber. So there's a lot more than rational to individualize this. And I would also, let's say the clinical effects were rather minor and perhaps also disappointing. So I think when it comes to dietary fibers, I have more hope in symbiotic probably approaches, and then probably also, you know, more complex one with mixtures of fiber, perhaps mixtures of microbes, and there I'm a bit more hopeful, and then also go for specific mechanistic targets to try and achieve something.
Yeah, sure.
So we've done some modeling of some of these prior intervention studies to try to understand, you know, responders, non-responders, from the microbiome metabolism context, and we definitely see a lot of variation there. And, you know, it does appear that, you know, you give 10 people a banana, very different chemicals come out the other end. So even for the exact same intervention, you're getting very different effects because of the microbiota. But I think we can target things. So if you could get to the point where you could pre-partition people into responders and non-responders to a given fiber, then you could just deploy different fibers to different people. To the mix question, we also find that if we mix fibers together in the models, we can reduce the average non-responder rate in the population. So mixtures, at least in the simulation context, appear to be more robust, and I think that fits too with like a complex diet being more robust. You're hedging your bets across a lot of different things. So whether you're doing something really difficult and complex for someone to adhere to possibly, or you're trying to do something more simple with a fiber mix, that might, or precision targeting.
Yeah, I think we talked about personalized nutrition, personalized medicine. Here, I think we should not forget personalized microbiome therapy. Because as many clinical trials already show, the baseline microbiota factors actually determine the microbiome targeted or dietary targeted or probiotics-based approaches. And so how these probiotics or with the prebiotics, not how we help these species can be colonization in your gut is a big question as well, because most of the prebiotic trials doesn't really have a long-lasting effect. So because it cannot colonize, so then we have to need more evidence to have a more ecological point of knowledge and a basis to help what is the most efficient, long-lasting effect we can achieve.
Okay. So diets, personalized diets. So if you would have to name the 3 foods that are the most important to drive changes that are beneficial and can help to improve healthy aging, what would there be?
Red wine.
Well, that's a fermented food. Something that hasn't come up in this discussion yet is fermented food, which is one of the original ties to longevity with yogurt. I don't know how much people have looked into fermented foods in the context of longevity, but that seems to be something that also increases diversity, possibly through the metabolites that are in there, also possibly delivers probiotics as well.
Yeah, fermentation in general, for dairy product in particular, has been shown to reduce the risk of type 2 diabetes and other related disease. And of course, all these chronic diseases are linked to aging. So, but I agree with you. I don't think I've seen really good studies that have looked in that context. Really.
So I think there's a lot of excitement around it, and in the nutrition field and in Cork we have several groups looking in it. But when you then look at the actual, human trials data, there's still very, very little. But I think this is hopefully going to happen now. Yeah.
But now fermentation, fermenting carbohydrate is good. Is it the same for proteins? Because I've seen a lot of presentation today at this symposium and others talking about some fermentation of some proteins will lead at least to the formation of maybe metabolites we don't really want. And it's very debatable. I think, Sean, you talk about phosphocrystal sulfate. Is it good or bad? It's same for TMAO, actually. There's a lot of debate. Yeah. So is fermentation good for all type of food or not?
Though I do not really know fermented meat products good or not, because I don't think we had much scientific evidence yet. But I'm really happy you raised the questions regarding the protein, because I think we have a lot of studies talking about the fiber. It seems, but I think they already can see that actually diet protein and studies in terms of the human health has been under-evaluated, because a lot of products metabolize relate actually amino acids metabolism, like tryptophan and tamale, that is a carnitine, and also arginine. They're all linked to all kinds of products, actually linked to all kinds of diseases. So all related to the protein intake, branch amino acids, a lot of amino acids metabolism. So I really think this is a big area. We should learn it even more. In addition, short-chain fatty acids.
And there's quite some controversy in this, actually. When we wrote our review on healthy eating in the microbiome, one of the authors is Carla Brado. She's from Brazil. It's very hard to convince a Brazilian, that meat is detrimental for you. Yeah, so there was a lot of, that was actually the one point where we had the most discussions. And obviously there's a lot of benefits. Actually, you mentioned it, I think, then and you have to, it's very context. It's very hard to make some strict, points, because then if you get into a frail, and that's Carla works on sarcopenia, you don't want to recommend low protein diets, to these individuals. But then in a longer term and throughout life, I think high protein diets are, at least that's what the nutritionist thinks, are more detrimental. And in mouse studies also, they do shorten life. But it's very, very important to have nuance there.
Yeah, it's also like with now the foodomics become more a rising area, right? Because also we are human beings. If you said we only eat one type of food, probably most of us say, no, I cannot live with that. I would rather enjoy it from life. So like a food diversity is a lot of component, right? You should actually, like we had the microbiome diversity, maybe you should take also a large variety of the food to enrich your diversity and enrich your metabolism and all kinds of the compounds. So not separate. Yeah.
And I still want to do a Korean barbecue tonight.
Yes.
With kimchi then.
Yes. We also want enjoying our life as well. The happiness from food is also important for healthy aging.
And that's,
I think from the, sorry.
No, I was going to say, that's one of the clearest results you showed, Jens, is that red meat is really bad. And there's all these people trying to push the carnivore diet out there, these influencers. Yeah. And I think that's just terrible for you. But it is complicated. Like we see, indole is kind of maybe good for you. It's anti-inflammatory and it's derived from tryptophan. But then there's a lot of others that seem to be bad. If you're constipated, you seem to have more of these. So it looks like there's a switch in the gut. Like normally your microbes will produce short-chain fatty acids from carbohydrates. But if it's a hot potato, right? If it sits around for too long, it runs out of substrate and starts to ferment proteins, and then you get this buildup. And that's been associated with chronic diseases like kidney disease or dementias. And then I've seen some evidence from trials where in young people, having high protein maybe is bad for you, but it really depends on if it's plant-based or animal-based. But if you do then that same intervention in like an older, frail population, suddenly you see benefits because you're protecting people from sarcopenia.
And I think the discussion about proteins is always low versus high protein diets. I think we don't think enough about looking at the composition or the different type of proteins in our diet for just a regular amount of protein in our diet. I think it's as important as to look at different type of sugar or of course lipid in the diet. I see there's only 1 minute left, so one question I have quickly for you guys. Most of the talk today were about the gut microbiome, but is there surrogate regions that we can use? What about the oral microbiome? Some of you touch about it. Some would talk about the skin microbiome. So sometimes these low-hanging fruit type of sites can be also very important. So what are your take on this in maybe 10 seconds for each of you? That's okay, 15 seconds then.
Yeah, so I'll take my 10 seconds. The answer is that these extraintestinal microbiomes are exceedingly interesting. These are niches in which the microbiome in each niche has co-evolved to generate functions that are unique to the niche. They're more complicated to study. They're usually lower biomass. The skin is one example. But also present opportunities to explore niche-related diseases that are neglected if we just look at the gut. So we and others are doing this extensively, but it's easier said than done in some cases.
And my second is that all areas, of course, important regarding aging. I think microbiome translocation across body side will be very important because skin microbiome can easily enter your mouth, and the mouth can be translocated to your gut. And so this transmission, how they migrated, how they at a different niche, what function they're going to play, whether they cause disease or not, I think it's a dynamic map of a microbiome that will be very interesting to investigate.
So the oral microbiome, when it relates to diet, is already important because of dental disease, which obviously is driven by sugar content in the diet, and then that has downstream effects. So it is obviously, we are not looking at it, but it is an important.
But it's relevant, yeah.
Yeah, I agree. I mean, it's the oral microbiome has changed a lot with the Neolithic Revolution, and it's created a lot of problems for our ancestors, and probably continues to do so for us today. It's definitely a health and diet readout.
Thank you.
I'll probably keep focusing on the gut, but I am excited about immunomodulatory effects in other compartments. And there's tantalizing work from the Fischbach lab right in mice that you can affect systemic immunity just by painting a microbe on the skin. So I think that work's really fascinating.
All right, thanks you guys for staying on time. And so that closes the symposium today. I would like to thank again the Danone Institute for sponsoring this event, all the speakers and the participants, of course, to the roundtable. And thanks to you for showing up and asking great questions as well. Thank you.
Thank you.